All right, I'm Nathaniel Polley, and I began my PhD approximately 2 weeks ago, I believe. And I will be defending today my thesis. If that works with you, Bruno. I have a restaurant to open, so I'm kind of in a hurry here. All right, so in all seriousness, I was an engineer d'études for Jean-Manuel Sarri's team, so I was providing bioinformatics support on different genomic-based projects. And when you look at a disease such as AML, hemopoietic related disease, that is very hematopoietic because it doesn't follow a specific narrative. You have to be both a jazz musician and a data scientist to be able to make sense of it all. So what I have to introduce to you to today is really just an agglomeration of my work for these projects, starting from survival prediction, going all the way to mechanism of action identification from human flux data. So I know I promised all of you machine learning, but I want to start off with a bit of history first. This is Abraham Wald. He was Jewish Austrian during World War II, and he fled to America to become a statistics professor. And he was so skilled in his craft that the US Air Force actually asked a favor of him. So this is one of the bombers that the US Air Force had. And he was tasked with the objective of where should we put the metal reinforcements on the plane to prevent them from crashing? So based upon where the bullets are, who would think that the reinforcements should go on there? Raise of hands. Okay, it's a very skilled crowd here, because the truth of the matter is that those are the planes that survived during the war. We were only looking at the survivors in this analysis. What we actually wanted to look at was where the bullet holes weren't, because those were on the planes that crashed. So this is an example of survivorship bias, where we're only looking at the results that have produced some sort of beneficial lesson or message. And that's the truth with most of all genetic research. It's always the title, gene XYZ has some sort of promise in acute myeloid leukemia as a new beneficial target. However, what that journal does not include is all the hundreds, if not thousands, of failures in the laboratory experiments that project had. So, which is also very critical information that cannot be overlooked. So, can we go to the literature to find novel mechanisms of action that hadn't been considered before, just by survivorship bias alone, not even taking into account human error or human bias? So we turn to in vivo experimental design, where we xenographed cancer cells into mice, breed them in the laboratory, and then run various tests on them. So we have to establish some sort of equivalence to the animal subject, to the ensemble, to the entire AML population for this to be a valid approach. So, there we are. In order to do this, we need to look at something known as the ergodic assumption. That's where we take one data point, which happens to be the mouse here, and over time it will have a moving average of a particular metabolite in a given place. We also have the entire human ensemble, a sample size, and then they also have an average for each sample. For it to be ergodic, these two have to be equivalent. A bit more illustrative example here. So that's the mathematical proof behind ergodicity. So this is a more three-dimensional view of the process, that you have each one of your sample traversing through a specific metabolite, specific state, and then the time average should be able to reflect all that in one organism. In a non-ergodic setting, you have these holes in the sample space of where the data has not been traversed. So in an ergodic setting, each sample has a chance of traversing all the space to reflect that of the ensemble population. So this right here is a picture of the human metabolism in Recon 1. So this is 3,000 reactions. Each dot is a metabolite, and each line connecting the dot is a reaction. This is actually the small model, the legacy model Recon 1, because this is what I had could fit on my memory space on the computer. However, the current model I'm using has 10,000 reactions, even bigger than that. It is quite a miracle that the human cell has to make trillions of correct actions a day in order for the human not to die. And certain members of parliament in my country struggle to make a correct decision in their entire career. I'm sure it's just perfect over here though, but politics aside. So this could be more easily represented on a two-dimensional plane surface where reactions that are more enriched, more active, can be shown. That's the red here as elevated, and those that are deactivated or going in the reverse direction will be blue. So we can establish that the sample space is a lot more dynamic than just one metabolite that is the course of the illness, as we see as the case with many bacterial and viral illnesses. We just have a few things to keep track of. However, in cancer, it's much more dynamic process. So if we take each one of these planes that could exist across an entire trajectory of time, we know that there's many different fluctuations that can occur. And then we also have to look at the ensemble average of the planes of the trajectory as well. So for the ergodic approach to be valid in this case, the cross sections of each one of these models have to be relatively equivalent. As in, if we take the average across each one of these planes here, it's going to be arbitrarily more or less equivalent to the next. However, the case of that to happen is practically close to zero. It's not exactly zero, however, it is a very negligible number, which reminds me of a song. Kung fu va je t'aime. Anyone know that? Some Balavoine fans in here. So it's like putting a message into a bottle and throwing it into the sea and expecting someone to read it. However, the solution might be, okay, we'll just do more animal studies. And that means that we cast a bigger net into the sea. However, the holes in the same net are bigger, so we're really not getting anywhere with that. So long story short, might as well take your odds at the blackjack table at the casino in Ile de Ramier, because this type of system is not going to, one, be in an ergodic setting to draw that equivalence. And additionally, the probability that we will stumble across something by chance is relatively low, even if it was a perfect ergodic setting. So for my master's, I had to take this class on how fluids flow through pipes. I guess it's fluid dynamics. I would just call it pipes class. But I still don't know to this day why I had to take it. But because if my sink backs up, I still have to call the plumber. But anyway, there are different ways that water can flow. Either it's laminar or turbulent flow. Laminar, where each plane is its own gentle velocity. Turbulent flows, high discharge, lots of randomness, lots of mixing eddies in the water. So I was thinking that data differentiation among cellular progression. So we start at the leukemic stem cell at the top and we move to a more differentiated state. As that state progresses, we start to see a larger increase in turbulence of that data. And turbulence doesn't really have an exact definition. It's more randomness, dispersity, an increase in entropy, so to speak. So Reynolds, who came up with an equation to describe laminar versus turbulent flow. If the number is smaller, it's more laminar approach. If it's larger, it's turbulent. So I thought I could do the same thing, but with using data flow to describe this phenomenon. So we have omega being the total size of the entire sample space. It's like the width of the pipe. N being the known, that should be n squared, by the way, so the units cancel. But I was up late last night figuring this out. So the known values, V is how many bits of information change per given unit of time. H of X being your Shannon entropy, and time being the total progression. And then as this number increases, we have a more turbulent data set where it's harder to make predictions. So we want this to be as low as possible, which in theory would be at diagnosis. So any prediction that can be made from diagnosis is our best case scenario in terms of differentiating. So Reynolds used his name for the coefficient. I decided for the Nathaniel Polley coefficient, I'll use the Roy Lee Pratt logo because everyone's going to need a martini after this. So it was just so conveniently made. So in a nutshell, what we have here is a laminar flow starting from here to here, and then a more turbulent state at the end. So even though these 2 points, picture this being a smoke particle where we have the initial trajectory, we have a much higher chance of predicting its outcome, even if they are farther apart than a point originating in the turbulent state, even if those points are closer together. So that's what the current theory states. No need to memorize the equation for the exam. I came up with it a few days ago, so it still needs a bit of work. So what's the solution to all the problems that we face? Don't reinvent the wheel. Just realign it. Think back to the planes. We just had to turn the solution on its side in order to make sense of it all and to eventually win that war. Because wars are won not just with good engineering and good information, but a good understanding of that information is what really takes victory to the next level. So what we need to do from the start, when we're just doing our preliminary screens, looking for mechanisms of action, just get rid of the entire in vivo mouse model to begin with and look directly at the patient data set, because that's where all of the variability is going to be at. The unadulterated data will be present among each one of the sample spaces. So a while back, I wrote a algorithm. It's called PolyMORPHOS, Metabolic-Oriented Regression Predicting Human Overall Survival. And it takes three things at the beginning: RNAseq of any platform, starting metabolic conditions to be able to mimic human blood, as well as a metabolomic model of your choice, in this case, human GEM. So you combine all those together, and what it creates is calibrated metabolic fluxes, which are much easier to understand and much more accurate than testing, say, hundreds of gene signatures for glycolysis and picking the best ones, because this will take into account each of the expressed genes to create a calibrated model. So this creates a reduction of dimensionality to just be one two-dimensional plane, so to speak, of metabolic states that can be inferred across time. So what does this look like? It looks like a list of reactions that are listed by their importance value as predictors, as well as a tree-like structure, which was previously mentioned before, where each one of these decisions is based on a different flux amount, and then they will traverse a different direction of the tree depending on the patient's final state. And what we get is different types of metabolites. So in this case, it's iron exchange. In the second case, it's retinol exchange. And in the third case, for day 450, it's a prostaglandin. And as we see from the Kaplan-Meier plots, the distance in between the confidence intervals is different for each line. So the confidence intervals, when they don't overlap, that indicates a p-value of 0.05 in terms of survival difference. I'll go over that in my next seminar, having confidence in your intervals. So these are the accuracies for this model, which makes the most sense because day 50, day 150 is the closest to the diagnostic data. So it's going to be the most accurate, close to around 99% accuracy AUC. Day 300, it relatively gets a bit lower, and then day 450, still above 0.7 in terms of AUC, but the variability gets more and more as the time progresses. So people say, okay, this is useful in Nathaniel for survival, but what about the gene applications in our lab? What about current projects in our lab? So what will connect any feature expression, say a gene, isoform, protein, anything you can imagine, with the metabolic model of survival? And that's my next album cover when after I graduate. So here I present to you SORI, which stands for Simulation of Universal Reactions in Silico. So it's a bit of a paradigm shift here. The traditional paradigm was we initially were just comparing two expression states, high expression and low expression, subtracting the two with the t-test, and completely ignoring the rest of the bulk of the population at baseline. And I learned a new word 3 days ago, LUFOC. LUFOC. Am I saying it correctly? LUFOC. LUFOC. Okay. So I believe this to be fucking ridiculous idea. Because first of all, we're ignoring the bulk of the patients, and based upon everyone's experience, patient data points are rare. We should be taking as many as we possibly can, not just looking at the extremes. And two, what if it just so happens that the baseline expression of the gene is responsible for that mechanism of action of survival or morbidity, and any perturbations away from that average would cause an increase that is of significance. It would be ignored if the effect was just canceled out with subtraction on both sides. So in SORI, we now divide the patients into low, baseline, and high expression based upon any given expression value of your choice, 25th percentile to 75th in this case. And we then run hundreds of simulations from PolyMORPHOS. So we sample with replacement, 80-20 split. So it will be a different population being generated each time. So I'm trying to take the towel of data and just run it out completely so that I get every single drop that I possibly can out of this data set. And then what I do is from the selected lists of reactions that are relevant to survival or death at each state, I then compare low versus high, baseline versus high, and low versus baseline in each risk group. So I'm looking at everyone in the population now. And then from this, we will get their own personalized maps of mechanisms of action in the metabolism that are relevantly distributed among the candidates. And then the new model that we have is not just high and low, good and bad, but for each state of baseline, low and high, we get survival risk, survival risk, survival risk. So we now have 6 different parameters that we can look up. So I have a new way to remember this. Say low, say no to high versus low. Dites au revoir à l'autre contre le vent. Look, rhymes in every language, like the Aribo slogan. So you have to look at the top, baseline, and bottom of all of your data sets, not just the extremes. So, old paradigm, new paradigm, much more insight on the activity of your metabolomic fluxes. So, this is one of the outputs of SORI. I will call it a polygraph so you know it's not lying. So this is the distribution of, say, the PSAT1 transaminase. So this gene specifically is to convert glutamate to alpha-ketoglutarate. All of them are to alpha-ketoglutarate, but I believe this one's glutamate. So this is your expression levels throughout the entire patient population, and each of the vertical divisions is the risk threshold, not risk, but expression threshold, high, baseline, and low. And what we have below is the results of the XGBoost output, which are in line with the x-axis. And this represents the activity of the particular action in question. So the more purple it is, the more active it is in the forward direction. The more yellow it is, the more it is in the reverse direction. And this is the reaction that was one of the reactions that was chosen by the software. So this is dimethylglycine from the cytosol moving in the forward direction to dimethylglycine in the mitochondria. So what we can infer from this is a connection between survival, gene expression, as well as metabolomic activity. And what we can conclude is that at the low expression of PSAT1 gene, we get a higher activity of DMGTM, dimethylglycine transport. And then as a result, this line shows that because it's above 0, it's predicting survival among these patients. Now look what happens when we drop over to the baseline area. We start to get a dip in this survival prediction. So in this case, baseline is the risk group. And if we were only comparing high versus low, we would completely miss this information, because as the gene gets higher, it relatively tapers off close to neutral, where we get this difference in survival prediction based upon the expression of this gene based upon this metabolite. It's the Rosetta Stone of sorts of computational biology. So based upon whether or not that reaction contributed to death and whether or not that gene state corresponded to that reaction, we now get an idea of different moods, metabolic moods this way, and metabolic personalities in this direction. So for instance, BCAT2, another transaminase, and the reactions over here that are red, these are the ones that contributed to death. These are the ones that contributed to survival. However, when we get to low BCAT2, which were the 2 most extreme differences here, it's inverted, meaning that red is where the blue was on the high BCAT, and blue was where the red is at the low BCAT. The baseline BCAT, however, which is here, baseline BCAT2, it's completely blue all the way around. So the best option in this case is not to completely eliminate the gene, get it into the low territory, but to neutralize it into the baseline territory as it's connected to most of the metabolomic outcomes. So for now, these are some of the different genes that my group is doing in high baseline and low groups. And then in this section here seem to be the commonality among all of the different states. And each one seems to have its own jurisdiction that is unique to its own. So it's like its own prefecture. And then this is the central hexagon here. And each one is its own prefecture of its different metabolomic states. So if we zoom in further to that, these are all the common mechanisms of action that have the most extreme differences among the states. And then I derived from that a list of the metabolites that are responsible for this. Now, can I get some noise on some mechanism of action here? Excited? Okay. All right, it's going to be a historic day because I'm not touching a single mouse or a single cell culture to get this information. I'm going directly from the patient data. So let's begin. Porphobilinogen, right? It's one of the precursors of the heme synthesis that is being generated here in the mitochondria. So heme is necessary in the blood to carry oxygen, also necessary to carry out other cellular phenomena. Look closely here. The precursor metabolites are glycine and succinyl-CoA. So we need those to be able to begin this process. So we have dimethylglycine, a precursor to glycine, hydroxypyruvate glycerate, which in turning to glyoxate pyruvate, it produces glycine and alanine. Serine and threonine also implicated in glycine metabolism. Now, gamma-linoleuco-CoA. So this tells us what the heme group is actually doing. So we have the cytochrome B5 with the iron in the heme group. And then as that is being oxidized, the linoleuco-CoA is turning into the gamma-linoleuco-CoA. And that is where we can see a functional target occurring. And this is relatively the intersection of everything I said above, from CDP to CMP. So this is the central location of it all. And then dimethylallyl isopentyl diphosphate. That is this backdoor approach right here, that once the heme is generated from heme B, it can then be personalized to heme O or heme A, given these metabolites here. So everything is pointing to heme metabolism in terms of mitochondrial function as mechanisms of reaction of resistance in acute myeloid leukemia patients. Not a single mouse had to die for this. Not any cells had to be worked on for this. It was all within an afternoon. We were able to get this information. So I will leave you with a quote. If the brain were so simple, we could understand it, then we would be so simple that we couldn't. So I know it's very moving, but it's easy in this world to slam the door in the face of new ideas and be afraid of the future. And I would like to thank the Care Graduate School, all the personnel and faculty for giving this opportunity a chance and having courageousness and openness to experience and curiosity. And I'd like to also thank everyone in the audience right now for being a part of this revolution, where we can get the mechanisms of action that we need faster so that we can get to cures faster and completely revolutionize the way medicine is formed. Thank you.